another one of the big ideas is the idea of microbes. And of course, the HSC course starts with the talking about Marshall and Warren and their journey to show H. pylori caused a majority of peptic ulcers. And then it becomes an important part as well in Module 8, talking about the impact of science. And of course, the development of smallpox vaccine with Edward Jenner, antimicrobial drugs, and vaccine programs to eradicate disease. And so in this case here, again, another area where there is a lot of data out there as well, historical data, particularly on the eradication of smallpox. And so it's something that is good, really good for writing questions as well, and particularly longer questions. And so you don't necessarily need to know the entire theory behind immunization because it can get a bit complex. But in terms of just the main idea that a pathogen or an antibody or an mRNA sequence is injected into the body, which triggers an immune response, which generates memory cells, which makes you immune. And that's basically what you need to know. The other thing as well is, as we said there before, there's a huge amount of data on smallpox, and you go from 1960 here with the data, and this is the World Health Organization data, where you had over 80 countries in 1960 saying they had cases. By the time you get to the mid-70s, and in 1980, the World Health Organization tells you that smallpox has been eradicated. And so in this case here, this is and part of the reason this is really good for questions is that it's a really good example of success, and that vaccine programs do work, and that they have positive impacts on society. Now, another one there, another cross-linkage, radioactivity, nuclear technology. And it goes across because we are talking in Module 6 about technology, and then in Module 8 we're talking about the impacts. And so this is a really good example of where the impact of a scientific idea can be linked directly to the technology. And there's a few things you just need to know that there, in Module 6 does specifically talk about detecting radiation, and you can talk about that most radiation is detected by a Geiger-Muller tube, or a Geiger counter, as it's commonly more known as. And look, you don't have to go into the whole idea of how it works, but it is a device that measures radioactivity. One of the biggest debates that comes out of nuclear technology is this idea of nuclear power. And particularly in the modern world, we are talking a lot about fossil fuels, alternative energies, and nuclear power is often promoted as something that could replace fossil fuels. And there are some advantages. There's no greenhouse gas emissions. It is fuel efficient. You only need a tiny amount of fuel to give a lot of energy. Now, resilient in times of extreme weather, yeah. Job intensive, definitely. Low operating costs, not so much. This graphic's a little bit out of date. If you look at the costs of power, nuclear is actually more expensive than coal now. And that's because the ability to actually operate and run and develop the plant is quite expensive. Now, of course, there are some new technologies coming on board which have the potential to be cheap. Thorium, small-scale reactors and that there, but they're also untested technology at this time. And of course, the cons, if you have a nuclear accident, it's bad, and devastatingly bad. Now, luckily, there hasn't been too many nuclear accidents, but when they've happened, they're catastrophic. Even outside of that, probably the most problematic part of nuclear power is the waste. And not all nuclear waste is long-term. Things like clothes used at reactors and that they're low-level waste, and eventually they will lose their radiation. But particularly nuclear fuel. And there is no solution at the moment on what to actually do with it. There's a lot of places around the world. There's no real cooperation worldwide. And we are, in some cases, talking about waste that will last for millions, if not billions of years. And that's really a potential future problem as well, that if society in 1,000, 10,000 years, information gets lost over time, and they may not actually realize that we've buried a whole heap of nuclear material under the ground or wherever. And there's also things America and Russia used to do, some fun things like just dump their nuclear waste in the ocean. And so there's barrels of nuclear waste in some of the world's oceans just sitting there decaying at the moment. However, nuclear technology is important. And even if you disregard power, we still need some nuclear technology. And so Australia has one nuclear reactor at Lucas Heights. It's a small reactor, it doesn't use much fuel, doesn't generate much waste, but it generates a huge amount of nuclear materials that are used in medicine. And at the moment, Australia produces a huge amount of the world's Technetium 99. And so stuff here is sent across the world. So, and that is positive for Australia as an export industry, but also positive for the people who are engaged in those technologies or in getting treated by that medicine. And which brings us to another 7-mark question here from the HSC. And this idea: describe how technologies had a significant impact on the development of atomic theory. And this is actually, it's a 3-mark question of a 7-mark question. And this is quite an open-ended question here, because you can talk about some different things here. A lot of people would probably talk about, particularly those who've got a bit of a physics background, you could talk about the cathode ray tube, which allowed people to understand the atom. So the cathode ray tube was able to determine that there were parts of the atom that weren't whole. So if you go back to ancient Greek theory, that the word atom obviously comes from the ancient Greek atomos, and that atom means indivisible. And then 2,000 odd years later, from the ancient Greeks, you get the idea of Dalton, and that atoms, again, are in tiny little spheres. And then you go to J.J. Thomson, who talks about the fact that those little spheres have little bits in them. And so in that case there, you talk about the cathode ray tube allowed scientists to understand that the atom, which they thought was indivisible, actually had smaller parts in it. And again, there's the example that the HSC uses, things like photographic plates. So you're talking about some of those early experiments, and the photographic plates showed that something was being given off by uranium ore. And so you're talking here about Becquerel and that idea there. But any technology, if you can talk about even further, if you want to get into some of the early 21st century experiments like Rutherford and things like that, you can link that in there. Any technology is linkable in this. And of course, how has it affected our understanding of radiotherapy? And this is the really interesting part here, because you do have to talk about the positives here. And so understanding of radioactivity has allowed us to develop new medical treatments that you can argue are non-invasive, so a person doesn't need surgery, and so that has minimal impacts. And if you look here as well, the sample answer is talking about that idea there, is that you can kill cancer cells without cutting someone open there, and it kills cancer. And you can talk about a few things there, but one of the things there, if you look at the 4 marks, talking about specific features of radioactivity and how that can help with radiotherapy. Another thing as well, and it's such a common misconception, do not talk about chemotherapy here. because chemotherapy is chemicals, that's injecting stuff in there. And if you look here, better responses, there's a huge amount of things here. You can talk about an immense range of different ideas across that question, whether it's a Large Hadron Collider, as I mentioned before, things like Rutherford's gold foil experiments and that there. But the key thing is to link the technology. And this is the thing here, when I say here, students with a holistic understanding will be advantaged, is this idea, it's just about understanding the things across a broad range. And so understanding the link between atomic theory and medicine and things like that. Another giant area that has a lot of reach across the course is DNA, biotechnology. And it comes up in Module 6 in terms of how DNA was discovered, in terms of X-ray diffraction, and Rosalind Franklin's image leading to Watson and Crick's discovery or finding on the shape of DNA. Also, genetically modified organisms as a technology that has come from our understanding of DNA. And then we get into Module 8, where in Module 6 we looked at just what the technology is, where it's led us, but how that impacts. And when we talk about genetically modifying sex cells and embryos, what are the impacts, and particularly what are the regulations? Same with bioweaponry. And there's also a little bit there in terms of cloning, stem cell research, all of this stuff there, and particularly GMOs makes a lot, is pretty heavy in there. And so in terms of X-ray crystallography, the main thing to understand is that you're firing X-rays at a crystal, and DNA and or any material, and you're looking at the patterns to work out what the shape of it is. And so this is what Rosalind Franklin was doing, and a whole heap of other people in that team as well. And then genetically modified organisms. So we're talking about you take a gene from one organism, insert it into another one so that you can have a new property in there. And in this case here, an example of disease resistance, drought tolerance, putting that in a tomato or something like that, and then generating or developing, growing a set of plants that are resistant to disease or drought or anything like that. And of course, one of the key things there, people do have a lot of fear of GMOs, but it's really important for students to realize that everything we eat is essentially a genetically modified organism. The difference being that that GMO process took place over 10, 12,000 years. We look at our farms. The main animals we have there, chickens, cows, sheep, pigs, goats, llamas, whatever, they have all been genetically modified over time. A cow is not found in nature. Things like bison, buffalo are, but they're very different. Sheep, aren't found in nature like that. They would die pretty quickly. Particularly you look at some of the Australian strains, like the Merino, and that there grows wool very quickly. Animals in nature don't grow hair that quickly, or they end up dying. Now, of course, as well, biological warfare. And one of the things to know about this one, it's banned. You also have toxins and things like that. And, some of these things do get developed in labs, but they're not allowed to be used. Anthrax is one that was common in America in the last 20 years. People were getting, because it's just a sort of microbe, and we're sending that. And the problem with anthrax is that it can lay dormant in a powder form, and then when it's released and you breathe it in, then it grows in your body. Bubonic plague is another one there. Smallpox, ricin, which is a sort of a pretty nasty one that's been used in terrorist attacks in Japan. And of course, one of the things there, people think this is a modern thing, but things like the bubonic plague back in the Middle Dark Ages, if you could fire a trebuchet with the body of someone with bubonic plague into a fortified area and spread the disease around there. World War One, mustard gas was being used in warfare in the trenches. And so what we have now is an agreement that countries are not going to use this. They're not going to use biological agents. And, there's been cases where alleged Russian spies have tried to kill people using nerve agents and that there. Well, of course, nothing's ever been proven. And of course, going back there to that idea of domestication, plant breeding, our modern farming techniques first started 10,000 years ago. And so you look at the domestication of wheat. And wheat is, of course, just a variant of grass. It's just that it's been selectively bred so that it's got bigger seeds. And then, by the time we get to Gregor Mendel, who did his pea plant experiments, we start working out we can crossbreed plants. And people have been doing that before that anyway. All that's happened now is that in the modern world, we can do that quickly. And so things like CRISPR technology can change genetic makeups very, very quickly. And that's the only thing that's changed from the old techniques, the speed at which we do it. As I said, there's 90% of the food in the world comes from 5 animals, and I think it's 12 plants. And they've all been genetically modified over the last 10, 12,000 years. Now, of course, we've got a 4-mark question from the HSC here, and simply what you have to do is explain how knowledge of DNA has led to 2 technologies. And so you just have to give 2 examples. And you can go anywhere with this. So the first thing is you have to describe the 2 technologies and then relate it to DNA. So you can talk about the Human Genome Project there as an example, where what we've done is we've mapped all the genes and developed a map, and how that has helped people discover things like where certain diseases are found on the genome. You can talk about cloning, CRISPR, genetically modified organisms. The sample answer gives you transgenic technology, which just that inserting of different genes in there, and forensics, where you can talk about DNA fingerprinting or anything there. The main thing here, and as you can see from the NESA response here, you talk about what the DNA structure is. You can talk about things like X-ray crystallography. You can bring that in there, but it's about saying how has the understanding of DNA, and each of that you have to mention DNA and link the DNA idea of DNA to the technology. And look, because that is such a big part of the syllabus, I think students really need to understand DNA and technology. It's one of those things that will be asked, I think, every year. And one of the other important cross-module things is this idea of Indigenous perspectives. And the course goes at it from 2 different angles. So in Module 6, we're just looking at some of the Indigenous ideas, and particularly this idea of medicinal plants, potential for drug treatments, and bioharvesting. So how we get this material from country. And so it's a really interesting area there, and there's plenty of examples you can use. And you can sort of delve into that a little bit, but I think more than anything else, just keep a couple of good examples. Maybe even one of, particularly when it talks about medicinal plants, at least 2 different plants that have medicinal benefits. And Module 8, because we are looking at the impact here, we're looking particularly starting off with this idea of protection of Indigenous ideas and cultural and intellectual property, and this idea of bioprospecting, and you can even bring in the idea of biopiracy, and how these traditional medical treatments can be used in the modern context. And so, as I said before, keep a couple of examples. Spinifex grass is used to help with skin problems. In the modern world, we blend it with coconut oil and stuff like that to make some nice soaps. The Dumbarumba, which is used for skin. You've got other things like the Gumby Gumby and that as well. And what you've also got here is this idea with bioprospecting of where people can come in and look at local people's knowledge, then have a look at it and see how can we do that. And in Australia, it's really important that that's done in cooperation. There's been countless examples in the past where traditional foods, traditional medicines have been sort of taken, and one of the biggest, well, two big examples. One is the fact that the macadamia nut is a native Australian plant, but plants were taken to places like Hawaii and South Africa, and South Africa now is the largest exporter of macadamia nuts. The other one is the Kakadu plum, which is a fruit found up in the northern part of Australia, which has the highest amount of vitamin C in any fruit ever discovered. And a little while ago, the American cosmetic company Mary Kay wanted to use Kakadu plum in a skin treatment, and in Australia, essentially legal action was taken to prevent them from doing that, and eventually Mary Kay sort of didn't go ahead with that. And last year there was one in 2020. Sorry, there was a multiple choice question on that there, and this idea of that this is about consultation, and so it's a good chance that each year there will be a written response on this, and particularly probably talking from that idea of how local people, how Indigenous people are included in these decision-making processes. Mining is another cross-module one in terms of in Module 7 and 8. And so in Module 7, we're talking about the idea of asbestos and cancer and lung cancer and how tragic that can be. And then in Module 8, we're talking about, well, first the ethics of mining practices and the idea that in Australia, all mining ventures have to have an environmental impact plan, and they also have to have a remediation plan so that after the mining has been exhausted from that area, the land is returned to its natural state as best as it can be. And so that's part of that. And that also impacts whether a mine is viable, because if they can only make a little bit of money out of it, then the rehabilitation costs may be more than what the value of the asset is there. And of course, the idea of petroleum and economic development. While we are looking at phasing as a world, as a species, looking at phasing out petroleum, it still is needed. We can't just go from using petroleum to none in a short period of time. And this idea here that economic development is often linked to it. So it's very hard in poorer countries to say just use electric cars, because electric cars are very expensive. And so it's about the idea of phasing and just being very cognizant of the fact that things sometimes have to take a little bit of time. And when we're talking about this here, so this idea of asbestos getting into the lungs, and asbestos being a mineral is very hard for the body to get rid of. And because the fibers are so small, they can sit in the lungs dormant for years, decades even, before they start having a big effect and you developing mesothelioma or asbestosis. There is also as well a newer one, which is silicosis, which is caused by people not wearing respiratory equipment when they're dealing with things like stone bench tops, which generate a lot of very, very fine dust, and that very fine dust gets into the lungs. And so silicosis is probably going to be an even bigger concern than asbestos, because silicosis happens very quickly. Asbestos usually affected people in their 40s or 50s. Silicosis can affect people in their 20s, so it can act a lot faster. And so we have a 4-mark question at a big thing here. And this idea of asbestos mining, and this idea particularly there, you've got a bit of information from the top there, and then the last part there saying that a report says that asbestos is safe and fibres will be dissolved in 2 weeks. And so part B here discussed the decision made by companies to continue to mine asbestos. Is this idea of, well, why would they continue to mine something that's inherently dangerous? And you can make some decisions there about talking about the fact that if safety procedures are put in place, then it's relatively safe, and there's a whole range of things you can put there as well. You can also go the other way as well and talk about that it's about disregarding the long-term health and miners in the industry, because again, so the counter argument each way is that while it is dangerous, some countries are poor in a lot of assets and a lot of resources, so they kind of have no choice. And so that's part of the problem with a lot of these, particularly societal impacts, is that in a lot of countries there's not much choice. And of course, one of the things there as well, you can talk about conflicts of interest, the fact that economics in a lot of cases overrides common sense and science because people just want more money. Again, pharmaceutical research is a huge area, and even though we do talk about things like placebos in Module 7, regulation, how tests are done, and ethics, these ideas do go across to other things. You might talk about an investigation from, say, Module 5 or 6, and then there might be ethical things there, and maybe there's double-blind trials involved with that as well. And of course, just the idea that double-blind trials is whether the researcher, if they knew what drug or what test was being applied, they may affect the outcome. I suppose you could almost link that to the Hawthorne effect as well. But of course, one of the main things about when we're talking about particularly pharmaceutical tests is whether people know the potential outcomes. And so it's considered, well, it is unethical if you ask someone to test a drug and you only outline the positive potentials. So you say, oh no, this will make you feel better, but you don't talk about other things, whether it's going to give them stomach cramps, whether it's going to do, give them nosebleed, like whatever the negative things there. So anyone who goes into a clinical trial should know all the potential outcomes, positive and negative. And this one here, simple question from the 2020 paper here. In a double-blind trial, do they know? And it should be patients don't know, researchers don't know. You also have there question 25 about recommendations for doing this here. And so in this case here, most of you would probably recognize that this is a generic version of the Marshall and Warren type thing here. But if Marshall and Warren did go to an ethics committee, what recommendations would they be likely to make? And of course, the story of Marshall and Warren means that Barry Marshall just sort of went a bit off the beaten track in terms of science. But the ethics committee would just talk about that you do follow the normal path of testing on mammals, things like rats, mice, just to make sure that there's no severe impact on mammals in general, and the idea of welfare and that there. And so one of the things that all trials usually take years and years to take, because it's a very slow process, because you don't just want people to test drugs on people in general, because that leads to negative outcomes. And of course, one of the things here, when you're talking about ethical investigations, how do you ascertain validity? How do you maintain reliability? And any of those, anything you can bring into that is good. Now, one little basic IS strategy here, and this is and this doesn't just apply to investigating science, it applies to all HSC things here. Don't leave blank spaces on your page. You look at the marking guidelines, and pretty much every marking guideline, if it's about 4 marks and above, or even 3 marks and above, the 1 mark response is any relevant information. And so if you put something down there that's relevant to the question, even if you're not sure about the question, you're still potentially going to get a mark, and 1 is better than nothing. Also, you might want to plan your answers. So particularly for 7-mark questions, or even 4 to 5, 6-mark questions, you might want to do a bit of planning. Put down some subheadings. Some of the examples we looked at briefly before. Depack the stimulus, right? Paraphrase what the stimulus is saying. And, you won't get all the marks, but you might get some marks. You might go above that. So if it's a 7-mark question and you've got no idea what's going on, maybe you might get 4 or 5 marks if you deconstruct the stimulus and put down some dot points of it. Maybe if there's a scientific investigation question, if you're not sure, maybe do a flow chart to show where it's going from one thing to the other. And of course, of those 5 things there as well, just sort of summing everything up. Again, all the way back at the start, the first 10 multiple choice questions will be fairly straightforward. The second set of 10 will involve a lot more analysing, a lot more critical thinking. So if you don't want to get bogged down, really good strategy, do probably the first 10, do the short answer, then come back. Know the terms validity, reliability, accuracy. You might have to apply them to a lot of situations. So if you're not sure about that, go back to every investigation you've done and apply them to it. Look for your study on the interconnected ideas, because they're more likely to be those extended questions. Practice extracting information from stimuli. Get a random graph. Write down what that graph means. And of course, don't leave blank spaces.